am Dr. Uma Maheshwari, Professor, Department of Physiology, PhD, IMS and R, Coimbatore. Now you have chosen to study about the regulation of acid-base balance by the body. Now maintenance of body fluids is important for the body enzymes to function normally. So decrease in pH known as acidosis will depress the neurons resulting in coma, whereas increase in pH known as alkalosis will increase the excitability of the neurons resulting in convulsions and cardiac arrhythmias. So the objective of this module is to make you know the importance of maintenance of pH of the body fluids within the normal range, to make you learn the mechanisms involved in the maintenance of normal pH, and to make you understand the complications that would arise if the pH is altered. Now pH, as you know, is defined as the negative logarithm of hydrogen ion concentration. The normal hydrogen ion concentration is very less. It's in the order of 0.07 zeros followed by 4 equivalents per liter. Precise regulation of the hydrogen ion concentration is very important because the activity of the various enzymes in the body are influenced by the hydrogen ion concentration. The normal pH of arterial blood is 7.4 and that of venous blood is 7.35. A person is considered to have acidosis when the pH falls below 7.38 and alkalosis when the pH goes above 7.42. Now survival is unlikely if the pH goes below 6.8 or if it goes above 8.0. Now let's see what is an acid, base and buffer. Acids, as you know, are proton donors. Now here hydrochloric acid is a strong acid because it can dissociate completely and donate the hydrogen ion, whereas carbonic acid is a weak acid because the reaction is reversible and it dissociates incompletely. A base is a proton acceptor. In this reaction, bicarbonate ion, hydrogen phosphate and hydroxyl ion are all bases because they can accept hydrogen ion. A buffer is a solution that resists the change in the pH of the solution following the addition of an acid or alkali. Now the dissociation constant of an acid is that pH at which the protonated and unprotonated forms are present in equal concentrations. A weak acid is most effective as a buffer when the pH of the solution remains close to the pKa of the acid. Now the second factor that determines the buffering effectiveness of the weak acid is the total concentration of the weak acid in the solution. Now because the concentration of hydrogen ion is very less, it is usually expressed on the logarithmic scale using the pH units as pH is equal to log 1 by hydrogen ion concentration, or you can also write it as negative logarithm of hydrogen ion concentration. So pH can be written as pH is equal to negative log 0.70 followed by 4, or pH is equal to log of 0.70 followed by 4 is equal to 7.4. That's how you get pH of blood to be 7.4. Now next we shall see the threats to the pH that alter the pH. Now, small quantities of acid or base that is present in the food and medicines and acids that are formed as a result of metabolic processes can alter the pH of blood if these are not removed from the body, they can alter the pH. But this is prevented by 3 primary systems that regulate the hydrogen ion concentration in the body fluids. Now they are the chemical acid base buffer systems of the body fluids, the respiratory system and the kidneys. So whenever there is a change in the hydrogen ion concentration, the buffer systems of the body fluids react within a fraction of a second to minimize the pH changes. The buffer systems keep them tied up until such time till the normal pH is re-established. Now the second line of defense is the respiratory system. Now the lungs act within few minutes to eliminate the carbon dioxide and therefore bicarbonate from the body. Now the third line of defense are the kidneys. Although they are slow to respond, it is the most powerful of the acid-base regulatory systems. Now coming to the bicarbonate buffer system. Now the bicarbonate buffer system consists of two components, namely the weak acid, carbonic acid, and its salt, sodium bicarbonate. Now carbon dioxide reacts with water to form carbonic acid in the presence of carbonic anhydrase, which being highly unstable dissociates to form hydrogen ion and bicarbonate ion. So you can see the reaction, carbon dioxide reacting with water in the slide, which forms carbonic acid, and this being highly unstable immediately dissociates into hydrogen ion and bicarbonate ion. The salt of carbonic acid is sodium bicarbonate, which dissociates into sodium ion and bicarbonate ion. Now when a strong acid like hydrochloric acid is added, it will result in the formation of hydrogen ion, which will be buffered by bicarbonate ion, resulting in the formation of a weak acid, thus preventing gross change in the pH. So you can see the reaction in the slide, that is hydrogen ion combining with bicarbonate ion giving rise to the weak acid carbonic acid, which immediately dissociates into carbon dioxide and water. Now when a strong base like sodium hydroxide is added, it produces opposite reaction. Sodium hydroxide will combine with carbonic acid to form sodium bicarbonate and water. So now a strong base is converted into weak base sodium bicarbonate, and thereby the pH change in the blood is prevented. Now we shall see the Henderson-Hasselbalch reaction for bicarbonate and carbonic acid. The law of mass reaction for this reaction is, now carbonic acid dissociates into hydrogen ion and bicarbonate ion. The pH is equal to pKa plus log bicarbonate ion divided by carbonic acid. So pH is equal to pKa plus log bicarbonate divided by concentration of carbon dioxide. Now according to Henry's law, the concentration is directly proportional to its partial pressure, and hence the above reaction can be written as concentration of carbon dioxide is equal to 0.03 into partial pressure of carbon dioxide. So hydrogen ion is equal to pK into 0.03 into partial pressure of carbon dioxide, where 0.03 is a solubility coefficient for carbon dioxide. Now substituting the values, we get pH is equal to 6.1 plus log 24 divided by 0.03 into 40. From the above equation, we can see that increase in bicarbonate concentration will result in alkalosis, and increase in PCO2 will result in acidosis. Now the bicarbonate concentration in the blood is regulated by the lungs. When one of these control systems functions are impaired, the concentration bicarbonate and carbon dioxide are altered, resulting in the alteration of the pH of blood. Now the bicarbonate buffer system is considered as the most powerful extracellular buffer in the body, as the two components of the buffer systems, namely bicarbonate and carbon dioxide, are precisely regulated by the kidneys and the lungs. Next, we shall see the role of phosphate buffer in the maintenance of pH of the body fluids. Phosphate is an important buffer in the intracellular fluid and also in the renal tubular fluid. Now the pK for this system is 6.8, which is close to normal pH, and hence it allows the system to operate near its maximum buffering power. Now the components of the buffer system is H2PO4- and HPO4-2-. When hydrochloric acid, which is a strong acid, is added to a mixture of these two substances, hydrogen ion is accepted by the base HPO4-2- and is ultimately converted into H2PO4, a weak acid. Now, so you can see the reaction: hydrochloric acid combining with Na2HPO4 forming sodium dihydrogen phosphate and sodium chloride. When a strong base like sodium hydroxide is added to the buffer system, the hydroxyl ion is buffered by the H2PO4- to form Na2HPO4, a weak base. So it is also an important buffer in the tubular fluids of the kidney, because it is becomes concentrated in the renal tubular fluid, and the pH of the tubular fluid is lower than the pH of the ECF, bringing the operating range of the buffer close to the pK 6.8. It is also an important intracellular buffer, as the concentration of the phosphate ion in the ICF is many times more than that in the ECF. Now coming to proteins are the most important buffers within the cells, as their carboxyl and amino groups dissociate, as you are seeing in the slide. On addition of an alkali, the hydrogen ion will be donated by the RCOH carboxyl group, and on the addition of an acid, the amino group will accept the hydrogen ion, forming RNH3 ion. The next important body fluid buffer is hemoglobin. The buffering capacity of hemoglobin is due to the dissociation of the imidazole group of histidine residues. The buffering capacity of hemoglobin is said to be 6 times more than that of the plasma proteins. Now, deoxyhemoglobin is a weaker acid and has a better buffering capacity than oxyhemoglobin because of its dissociation capacity like this. So it is so the reaction, hydrogen ion combines with hemoglobin, forming HHB, which is known as reduced hemoglobin. Now let's see the role of the lungs in the regulation of acid-base balance. Now the lungs constitute the second line of defense against acid-base disturbances. Now whenever there is increase in ventilation, it eliminates the carbon dioxide from the ECF, which in turn reduces the hydrogen ion concentration, and decrease in ventilation increases the hydrogen ion concentration. So increase in the rate of metabolic formation of carbon dioxide will result in increase in PCO2, the ECF, and decrease in the metabolic formation of carbon dioxide will result in the decrease in PCO2 in the ECS. So increase in hydrogen ion in the ECF stimulates the alveolar ventilation by stimulating the respiratory central chemoreceptors that are present in the ventral aspect of the medulla, and cause a washout of carbon dioxide and thus increase in pH. Now decrease in alveolar ventilation will result in retention of carbon dioxide in the body and decrease in pH. Thus the respiratory system acts as a negative feedback controller of the hydrogen ion concentration. Now whenever there is impairment of lung function, it can cause buildup of carbon dioxide in the body, resulting in respiratory acidosis, and the compensatory mechanisms in case of metabolic acidosis are blunted. Now moving on to the renal regulation of acid-base balance. Now the kidneys constitute the third line of defense against acid-base disturbances, but they take few hours to respond, or even few hours to days to eliminate the excess acids or bases that are formed in the body. Now the amount of non-volatile acids formed in the body as a result of metabolism of proteins per day is 80 milliequivalents. Now the kidneys are the only route by which these non-volatile acids can be excreted from the body. In addition, the kidneys should also prevent the excretion of bicarbonate from the ECF, which is known as the primary buffer system of the ECF. The kidneys regulate the ECF hydrogen ion concentration basically by three mechanisms. One, by secreting hydrogen ion, and next by reabsorbing the filtered bicarbonate, and 3, by producing new bicarbonate. Now this slide shows the amount of bicarbonate ion that is filtered by the kidneys in one day, and the percentage of bicarbonate that is reabsorbed in each part of the nephron. Now the loss of bicarbonate is prevented by the secretion of hydrogen ion, which reacts with the filtered bicarbonate. Now let's see how the hydrogen ion is secreted in the proximal convoluted tubule. Now the thick ascending limb of the loop of Henle and the early distal convoluted tubule secrete hydrogen ion by secondary active transport, where the secretion of hydrogen ion is coupled with the transport of sodium into the cell. This sodium gradient is established by the sodium potassium pump located in the basolateral membrane. More than 90% of the bicarbonate is absorbed in the PCT, requiring about 3,900 milliequivalents of hydrogen ion to be secreted. Now the carbon dioxide that diffuses into the tubular epithelial cells, or that is formed by the metabolism within the epithelial cell, reacts in the presence of carbonic anhydrase to form carbonic acid, which immediately dissociates into carbon dioxide and water. Now the hydrogen ion is secreted in the tubular fluid by sodium hydrogen counter transport. Sodium moves into the cell by a concentration gradient provided by the sodium potassium pump present in the basolateral membrane. The bicarbonate formed in the cells moves downhill across the basolateral membrane into the renal, interstitial, capillary blood. For every hydrogen ion secreted into the tubular fluid, a bicarbonate ion enters into the blood. Now the bicarbonate that is filtered by the glomerular capillaries reacts with the secreted hydrogen ion and forms carbonic acid. Now this reaction is catalyzed by carbonic anhydrase produced by the brush border of the tubular epithelial cells. The carbonic acid formed immediately dissociates into carbon dioxide and water. The carbon dioxide diffuses into the apical membrane of the epithelial cells and enters the cell, where it reacts with water forming carbonic acid, which being unstable immediately dissociates to form bicarbonate ion and hydrogen ion. The bicarbonate is transported by sodium bicarbonate and chloride bicarbonate exchanger. For each hydrogen ion formed in the tubular epithelial cell, a bicarbonate is also formed and is released back into the blood. So starting from late DCT, that is distal convoluted tubule, and the remainder of the tubular system, the tubular epithelium secrete hydrogen ion by primary active transport. So in this part of the nephron, it is not secondary active transport. The mechanism of secretion of hydrogen ion is by primary active transport, where the energy for pumping hydrogen ion is derived from the breakdown of ATP to ADP. Primary active secretion of hydrogen ion occurs in a special type of cell which is known as intercalated cell. The hydrogen ion secretion, the PCT, reduces the pH to only 6.7, whereas the hydrogen ion secretion in the collecting tubules can reduce the pH to 4.5, which is a lower limit of pH that can be achieved by the kidneys. So the amount of hydrogen ion secreted per day is about 4,400 milliequivalents per day, and the bicarbonate filtered is 4,320 milliequivalents per day. The slight excess of hydrogen ion in the tubules rids the body of the non-volatile acids, that is the fixed acids produced in the body, and in the process a new bicarbonate is generated. Next, moving on to the mechanism of generation of new bicarbonate ions by the kidney. So once all the bicarbonate ions are removed by the hydrogen ions, the phosphate buffer system carries the excess hydrogen ions into the urine and in the process generates a new bicarbonate. When there is excess hydrogen bicarbonate ion in the urine, they combine with buffers other than bicarbonate, and this results in the formation of new bicarbonate which enters the blood. So it is an important buffer in the tubular fluid. Because it becomes concentrated in the tubular fluid and because it is poorly absorbed. Now the pK of this system is about 6.8, which is close to pH of the blood. Once all the bicarbonate has been reabsorbed, excess of hydrogen ion will combine with hydrogen phosphate to form dihydrogen phosphate and is excreted as sodium hydrogen dihydrogen phosphate. The process of hydrogen ion secretion is same as described previously. Here the bicarbonate that is released in the blood is not the same bicarbonate that is reabsorbed, but instead it is a new bicarbonate and it is not the replacement of the filtered bicarbonate. So only 30 to 40 milliequivalents per day is available for buffering of excess hydrogen ion in the tubular fluid, and this excess occurs through ammonia buffer system in case of acidosis. Once all the hydrogen ions are buffered by bicarbonate and phosphate, the excess hydrogen ion is buffered by the ammonia buffer. Ammonium ion is synthesized from glutamine, which comes mainly from the metabolism of amino acids in the liver. The glutamine delivered to the kidneys is transported into the epithelial cells of the proximal tubule, thick ascending limb of loop of Henle, and distal convoluted tubule. Within the tubular epithelial cells, each molecule of glutamine is metabolized into 2 ammonium ion and 2 bicarbonate ions. The ammonium ion is secreted in the tubular lumen by counter-transport mechanism in exchange for sodium, which is reabsorbed. The bicarbonate ion is transported across the basolateral membrane along with the reabsorbed sodium into the interstitial fluid and is taken up by the peritubular capillaries. For each molecule of glutamine metabolized in the proximal tubule, 2 ammonium ions are secreted into the urine and bicarbonates are reabsorbed in the blood. The bicarbonate generated by this process constitutes what is known as the new bicarbonate. In the collecting tubule, the addition of ammonium ion to the tubular fluid occurs through a different mechanism. The hydrogen ion secreted by the tubular epithelial membrane into the lumen combines with ammonia to form ammonium ion, which is then excreted. The collecting ducts are permeable to ammonia gas, which can diffuse into the tubular lumen, but the luminal membrane is less permeable to ammonium ion, and therefore the ammonia reacts with hydrogen ion to form ammonium ion and is thus trapped in the tubular lumen and excreted in the urine. For each ammonium ion excreted, a new bicarbonate is generated and is added to the blood. In chronic acidosis, hydrogen ion is eliminated and is eliminated by excretion of ammonium ion. Now moving on to acid-base disorders. If the pH of the arterial blood is less than 7.4, the condition is called acidosis, and if the pH goes above 7.4, the condition is called alkalosis. Acidosis could be due to respiratory cause or metabolic, and alkalosis could be due to respiratory cause or metabolic cause. In metabolic acidosis, there is decrease in pH due to decreased excretion of hydrogen ion by the kidneys, and this is compensated by the respiratory system, which responds by increasing the rate of respiration. Respiratory acidosis is caused by decrease in the rate of respiration, which increases the partial pressure of carbon dioxide and hydrogen ion concentration, and this is compensated by the buffers of the body fluids and the kidneys, which respond by excreting an acidic urine. In metabolic alkalosis, there is increase in the plasma pH and a decrease in the hydrogen ion concentration. The increase in pH is due to increase in bicarbonate concentration. This is partly compensated by decrease in the respiratory rate, which increases the partial pressure of carbon dioxide. There is also an increase in the filtered load of bicarbonate, which fails to be reabsorbed and is excreted in the urine. In respiratory alkalosis, there is an increase in ECF pH and a decrease in hydrogen ion concentration. The cause of the alkalosis is a decrease in the plasma partial pressure of carbon dioxide caused by hyperventilation. The reduction in PCO2 leads to a decrease in the secretion of hydrogen ion concentration by the renal tubules. The decrease in hydrogen ion concentration reduces the amount of hydrogen ion in the renal tubular fluid. Hence, there is not enough hydrogen ion to react with all the bicarbonate that is filtered and is excreted in the urine. This results in the decrease in the plasma bicarbonate concentration and correction of the alkalosis. So to conclude, maintenance of pH is very important for the various body enzymes to function. Decrease in pH, known as acidosis, will depress the neurons resulting in coma, whereas increase in pH, known as alkalosis, will increase the excitability of the neurons resulting in convulsions.